Hello everyone. In last class we have discussed about the Griffith experiment, that is about transforming principle. There it was not clear about the biochemical responsible for transformation of rough strain of bacteria into smooth strain. Now in this class let us continue with the experiments which were conducted to prove DNA is a genetic material, that is biochemical characterization of transforming principle. Earlier it was thought that protein is responsible for the transformation of rough strain of bacteria into smooth strain. That is, it was considered as a genetic material. That means it is responsible for transformation in bacteria. But later on, scientists were having a doubt and they started doing some continuation experiments of Griffith. So, three scientists, namely Oswald Avery, Colin MacLeod and Maclean McCarthy. These three scientists have worked nearly for 10 years, that is starting from 1933 to 1944, to determine that the biochemical nature of transforming principle, that is whether it is DNA or protein or RNA. So earlier it was believed that protein is the genetic material. Now they have to prove, that is protein is the genetic material or something else present in a cell is responsible for transformation of rough strain of bacteria into smooth strain of bacteria. Now let us understand what these three scientists have did during the course of their experiment. You have to remember here, this experiment is a continuation of transforming principle which was conducted by Griffith. So let us take a quick look at what Griffith has found out of his series of experiments. In last step we have discussed already, S strain of bacteria which was heat killed was mixed with R strain of bacteria which was live. Together it was injected into mice and the mice was dead, and live S strain was recovered from the dead mice. And for this Griffith has concluded some transforming principle present in smooth strain of bacteria has transformed the R strain to secrete or synthesize a mucus coat and become virulent, and it caused death of the mice. So now the transforming principle could be protein, DNA or RNA, but that was not clear by Griffith experiment. Now what these three scientists, that is Oswald Avery, Colin MacLeod and Maclean McCarthy did is they took heat-killed S strain of bacteria and purified protein, DNA and RNA out of that bacteria. Now we can call this mixture as an extract. So that way we can say that they took the extract from heat-killed S strain of bacteria. Now this extract was first mixed with an enzyme called protease. You remember, proteases are the enzyme that are able to digest protein. So whenever there is a protein, it will break down into pieces. So protein will not be working over there. And this was mixed with R strain, which is live, and it was injected into mice. You remember here there is remaining only DNA and RNA. Now at this time the mice was dead. That means transformation has occurred. That is R strain was converted into S strain. So which means that protein is not responsible for transformation of S strain or strain of bacteria into S strain. That means in textbook they have given protease treatment with proteases did not causes any changes in transformation. That is, it did not affect transformation. In second step, what they did is they took a extract and it was mixed with enzyme RNase. So RNA is an enzyme, it is responsible for digesting the RNA. Then it was mixed with R strain of bacteria which was live. Again it was injected into mice and same result was obtained as before. That is, mice was dead. From dead mice there was extraction of S strain, live S strain. That means what? RNase did not affect transformation of R strain into S strain, which means RNA is not responsible for transformation. Later, the extract was mixed with DNase enzyme. DNases are the enzyme that are responsible for digesting DNA. Now this extract was mixed with R strain, live R strain, and then it was injected into mice. But now there was no transformation of R strain into S strain. That means the mice was live. Means what? R strain has not converted into S strain. That is, from this it is concluded that the treatment of the extract with DNases has inhibited the transformation. That means if there is no DNA is deleted or degraded in presence of DNases, hence there is no transformation of live R strain into S strain. Hence DNA is the genetic material responsible for transformation. Hence it is considered as DNA is the hereditary material. But at that time, all scientists or biologists were not convinced with this conclusion. Hence, they have to go for, other scientists have tried to prove DNA is the genetic material by other experiments that we shall look at in the upcoming videos. Now, in NCERT textbook, they have asked one question. Can you make the differences between DNA and DNases? So, here you go. DNA is the nucleic acid. You know that there are two nucleic acid, DNA and RNA, and DNA is responsible for storage of genetic information in majority of the living organisms except few virus. Whereas DNases are the enzymes, that is nothing but proteins, that you know that all enzymes are proteins that break down, that is DNases are the enzymes that break down DNA by cleaving the phosphodiester bond present between the nucleotides. Nucleotides are the building blocks of DNA. I hope you are all clear with this experiment, that is biochemical characterization. In next video, I'll come up with the other experiments. Thank you.